Ever wondered how cells regulate their cycle? This intricate process, known as cell cycle regulation, is a fascinating dance of biology happening in every cell of every living being. It's the process that allows cells to grow, divide, and create new cells in a controlled, organized manner. It's incredibly important because when cell cycle regulation goes awry, it can lead to serious conditions like cancer. A key component of this regulation process are proteins known as cyclins. Cyclins are like the conductors of an orchestra, guiding the musicians, or in this case cells, through each phase of their performance or cycle. These proteins ensure that each phase of the cell cycle occurs in the right order and at the right time, maintaining harmony within the cellular community. So these cyclins play a crucial role in regulating cell cycles, but how exactly do they do it? Stay tuned to find out. Cyclins are a group of proteins that are core regulators of the cell cycle. Acting as the conductors of the cellular symphony, these proteins orchestrate the various phases of the cell cycle. How do they do this? Through either activating cyclin-dependent kinases or sparking into action other enzymes or complexes. Now, let's delve a bit deeper. In humans, we have 4 different types of cyclins: G1 cyclins, G1/S cyclins, S cyclins, and M cyclins. Each of these cyclins have their own unique role regulating different phases of the cell cycle. So why are they called cyclins? Well, it's because their concentration in the cell fluctuates during the cell cycle, much like the waxing and waning of the moon. Their concentration remains low for the most part, but spikes dramatically when they are needed. These cyclins are named for their fluctuating concentrations in the cell during the cell cycle. The concentration of these cyclins usually remains low for the most part, but peaks dramatically when needed during the cycle. Sounds simple, right? But the mechanism of cyclins is a fascinating symphony of biological processes. Let's dive a bit deeper. The activation of cyclin proteins is stimulated by the binding of growth factors to the cell receptors. This binding action triggers the transcription of cyclin genes, essentially giving the green light for cyclin production to ramp up. But cyclins don't work alone. Most cyclin proteins act by forming a complex with cyclin-dependent kinases. Imagine a dance duo where one partner can't perform without the other. Together, they form a complex that takes center stage in the regulation of the cell cycle. However, not all cyclins are created equal. Take G1 cyclins, for instance. These particular proteins act as rate-limiting proteins for cell cycle progression. Think of them as the bouncers at the entrance of a club, controlling the pace of the party inside. By overexpressing the cyclin genes, G1 cyclins can speed up the transition of the G1 phase, effectively kicking the cell cycle into high gear. This brings us to an interesting point. Even though cyclins do not have any enzymatic activity on their own, they induce different processes in the cell cycle by providing binding sites for other enzymes. So while they may not be the life of the party, their role in the cell cycle is indispensable. They are the unseen conductors, orchestrating the rhythm and flow of cellular life. So, we've learned quite a bit about cyclins and cell cycle regulation, haven't we? Let's take a moment to revisit the key points we've discussed. Cyclins, as we now know, are a group of proteins that serve as core regulators in the cell cycle. They come into play during different phases, activating cyclin-dependent kinases or other enzymes and complexes to ensure the orderly progression of the cycle. We've also discovered that there are 4 primary types of cyclins in humans: G1 cyclins, G1/S cyclins, S cyclins, and M cyclins. Each type of cyclin regulates a different phase of the cell cycle, hence the specific names. The term cyclin itself is quite interesting, isn't it? Derived from their cyclical nature of concentration within the cells, these proteins usually maintain a low concentration but peak dramatically when needed during the cycle. We've delved into the mechanism of cyclins as well. We've learned that the activation of cyclin proteins is stimulated by the binding of growth factors to cell receptors, which in turn triggers the transcription of cyclin genes. Remember how some cyclins, like the G1 cyclin, act as rate-limiting proteins? They can accelerate the G1 transition by overexpressing cyclin genes, effectively setting the pace for cell cycle progression. Even though cyclins don't possess any enzymatic activity themselves, they facilitate various processes in the cell cycle by providing binding sites for other enzymes. Understanding these processes and how they work is key to understanding life at a cellular level. Until next time, keep learning and stay curious.